So before going into the methods for trajectory inference analysis, we need to talk about at least two new dimensionality reduction techniques that are quite often used for trajectory specifically. So one of them is ICA, independent component analysis, and that was already published in the first paper of Monocle, for example. The idea of ICA is a little bit different from PCA. The idea of ICA is that imagine that you have your true biological signals from this pseudo-time perspective of our cell differentiation, and there are several processes that are ongoing on the cell that we actually don't see. So for example, receptor signaling when you have the activation, and then the start of the cell activation per se, and then cell proliferation, and then expression of differentiation markers. So these are four processes that go on the cell that we usually don't really see per se, because what we actually see in the data is like the gene expression A, B, C, and they are quite mixed profiles, and they start at different time points. So what ICA does is try to take what is common across all those gene sets that you have and decompose them in independent components. So here you have, for example, this curve is present in many genes. This curve is present in a couple of genes. This other curve here in the middle is present in another genes. So what ICA tries to do is to decompose that variance into components that are independent of each other, like this. And in that way, you can define what is the expression trend for like this marker expression, for proliferation, for activation, for signaling. So comparing to PCA, PCA will try to find the highest variance in your data, and then the second highest variance for principal component 2. But for ICA, you will try to find these independent components, regardless of this angle here. So here for PCA, it needs to be 90 degrees. Here we'll try to find and fit these curves on your data like this. However, it assumes that the signals are independent of each other, which might not always be true. So cell activation, cell proliferation, cell signaling, or expression of some markers, they are all kind of dependent in molecularly. So it might not be always the best case to use ICA, but it is possible to use. And the values, one of the assumption is that it's not Gaussian distribution, but this is more technical detail. So yeah, ICA might fail in your data set, especially if you have a very big data set, to find these noise and signal in, to find the signals in your data. So ICA is a linear also method for dimensionality reduction. It estimates the sources that compose the data. So the idea is to take several genes and decompose into independent components. Yeah, so the components are assumed to be independent of each other, which might not be always true, but for simple data sets, it works pretty well. So some of the problems, he assumes that distribution is not Gaussian, which might not be true, and each component has equal importance. So the problem is that you need to know how many independent components you want to look for, because you can, like PCA, you can sort or filter them by importance, but for ICA, all of them will be important. So these are some of the problems with ICA. Do you have any questions right now? If you have already used Monocle, you probably have seen that before. No?
I have a question.
Yes.
So is it possible to do PCA before ICA to reduce, like to make some kind of variable selection before ICA?
Yes. Yeah, I think you can also do that, but the default would be do ICA on the normalized data, because then you have also all those mixed effects from each gene, and that will be the cleanest way, I think. But yeah, I think it is possible, yeah. The problem of doing on PCA is that you lose like what is the importance of each gene to that ICA, but rather you'll have the importance of each principal component to each independent component.
Yeah, right.
So, and ICA is also implemented in some packages, for example, Surat, I'm sure it is there. For ScanPy, I'm not sure, and for Scran is not there. but you can also run it independently as well.